Bandyopadhyay, Assistant Professor of Biology. Today we will discuss about enzymology. Enzyme, which is a biocatalyst, and enzymology is the study of enzymes. So enzymology is a branch of biochemistry that focuses on the study of enzymes which are mainly proteins. So there is one exception called ribozyme, which is made with RNA, but most of the enzymes are proteins. This acts as a biological catalyst, means the enzymes enhances the reaction of a particular, and for forming a reactant to products, enzymes increases the rate of the reaction by reducing the times. Enzyme accelerates the chemical reaction in living organisms, making them essential for processes such as digestion, DNA replication, transcription, metabolism, etc. And without enzymes, many biochemical reactions would occur in very slow rate. So enzymes do not start or end a particular reaction. It just enhances the rate. The tart enzyme was coined by Kuhne in 1878, and the first identified enzyme was diastase, amylase, pepsin, and then invertase. Properties of enzymes. We already told that the enzymes are biocatalysts. It's present in biological organisms and it is a catalyst. And then it lowers the activation energy. We will discuss that. Speed of the rate of the reaction. Enzymes are proteins. Mainly it is protein. Enzymes remains unchanged after reaction. After a reaction, though the reactant will be formed in the product, but enzymes will remain unchanged. Enzymes are specific to the substrate of a reaction, and enzymes are reversible. Changes in pH. Reversible and catalyze a reaction in both ways. Enzymes are denatured by change of temperature or change of pH. So, enzyme basically is a protein. So, if temperature or pH change, then enzyme will be denatured. Mechanism of enzymes. By understanding the mechanism of enzymes, we will have to understand two different properties. One is the models, one is the lock and key model. In lock and key model, it's told that the enzyme's active site has a specific shape that exactly fits the substrate. So, like a particular key is required for a particular lock, here also in enzyme there is an active site where a substrate binds for forming the product. So, in a particular specific active site shape, the particular substrate will bind. But another model is an induced fixed model. Here it is said that the enzyme's active site undergoes a conformational changes for binding the substrate. So, this we can see that this active site is not for this substrate. So, for binding it goes conformational changes. So, these are the enzyme substrate complex. So, these are the two different models. Enzyme classification. There are total 6 different types of enzymes. One, oxidoreductase. So, in there both oxidation and reduction takes place. Another one is transferase, which transport a particular group. Another one is hydrolysis, where water breakdowns. Here you can see H2O. So, OH, water breakdown. Lyase enzymes involve eliminating reaction of absence of water, leading to formation of double bonds. So, this is a lyase. Example, aldolase. Isomerase is an and which catalyzes the isomerization, means changing the structure, changing the form, and ligase, which is called as molecular glue, which is required for joining. So these are the factors which affects the enzyme, like pH, water, temperature, enzyme concentration. This is for enzyme concentration, some activators, some substrate concentration. And also there are some for enzyme inhibitors. There are some inhibitors which inhibits the action of the enzymes. Enzyme action. Why? How do enzymes minimizes the time of the reaction? By lowering the activation energy. So without enzyme, the activation energy will be like this. With enzyme, the activation energy will be like this. So it lowers the activation energy, so the time will take lesser. Enzyme kinetics. Just because enzyme is related with the rate of the reaction, so there is a kinetics. Based on that, there is a formed equation which is the Michaelis-Menten equation. And the Michaelis-Menten equation, there is different types. One is V0 or Vmax. V0 is the initial velocity and Vmax is the maximum velocity. S is the substrate concentration, Km is the Michaelis Menten constant. So this is the formula, V0 equals to Vmax into S divided by Km plus S. So here you can see this is the substrate concentration and this is the initial Vi or V0 is the initial velocity. So it is the maximum velocity and Km is the when the Vmax is half, means when the Vmax is half, then the substrate concentration is the Km. So Km is the substrate concentration when the Vmax is half. In enzymes, there are some inhibitors like competitive inhibitors, non-competitive inhibitors and uncompetitive inhibitors. Competitive inhibitors compete with the active site of the enzymes. So in competitive inhibitors which binds the active site, so the substrate can't bind. The non-competitive inhibitors bind the another site other than the active site, so the substrate can't bind. And uncompetitive inhibitors bind the enzyme substrate complex, so minimizes the rate of the reaction. So applications of enzymes, there are some enzymes which is required for medicinal production, there are some enzymes which are required for industrial purposes, and some also in the environmental like bioremediations or waste treatment, wastewater treatment, some enzymes are required.